Hello, I'm Gregor. I'm a PhD candidate at TU Wien, and I'm working at Biomotion Technologies here in Vienna. When the Berlin Wall fell in 1989, I wasn't born yet. But Diego, my co-founder, he was in first grade of elementary school in Mexico. As a child, I wanted to invent a machine that could produce whatever I wanted. Most of all, I wanted it to make edible books that would transfer the knowledge by eating them. Unfortunately, this wish has not come true yet. Our research is breaking the wall of bioprinting innovations. Bioprinting is similar to regular 3D printing, but deposits human cells and biomaterials with the aim of functionally recreating human tissues and organs. Human organs are complex tissues containing different types of cells, connective tissues, blood vessels, and much more. You see, in order to recreate such an organ using bioprinting, a lot of different printing techniques are required to replicate the single compartments. And some of these printing techniques haven't even been discovered yet. Bioprinting is still in its infancy, and the idea of whole 3D printed transplant organs is probably still decades away. While there have been a number of innovative developments in the field, many more are needed to make this dream of once printing a full organ a reality. However, one of the major restraints of innovative developments is that current machines don't allow the implementation of novel ideas. They're static by design. And this is precisely what we want to change. At Biomotion Technologies, we have built a development platform for 3D bioprinting that encourages users to develop novel strategies in the most user-friendly way possible. We've also built a software around it that will bring these new developments into a bioprinting routine to rapidly validate their benefits. Users have full control over every aspect of the device, so they can deploy and assess their developments in no time. We've developed a simplification layer for our bioprinting framework for users to freely develop without knowledge in programming or mechatronics. This approach has reduced the time of customization from 4 to 6 months down to 1 to 2 weeks, and thereby shortened innovation cycles significantly. Our modular approach will innovate on 3 sides. It will encourage scientific collaborations, as users can share printing technologies without the need to send custom-built printers as a whole. It enables the combination of established and novel techniques in a single process, and it increases the chance of scientific commercializations, as users do not need to maintain a whole machine with software, but just a module. This, in turn, also means that innovations are not buried in a drawer after publication. Bioprinting is still in its infancy. In the short term, we believe that the shortened development cycles will lead to innovations in tissue engineering, for drug testing, and personalized medicine. In the long term, we aim to develop an ecosystem of printing technologies that will enable off-the-shelf tissues for transplantation and thereby close the gap to organ fabrication and address the donor organ shortage. Which questions remain unanswered? On the one hand, we need to find ways to reach highly talented teams that do not have access to cutting-edge bioprinting technologies. And on the other hand, we need to find solutions to upscale the production of tissues and make them available for patients in hospitals.